welcome everyone, and thanks for joining us today for Neuroelectrics webinar. So this is the fourth of a series of webinars with a running title, Everything You Always Wanted to Know About tES, about transcranial electrical stimulation. I have a pleasure to co-host today's webinar, like always from our office in Barcelona, together with Laura Doubleblind, connecting from our office in Boston. Hello, Laura.
Hello, Rafael.
Today, we'll be talking about tES and EEG from home with telemedical research during COVID-19 and beyond. We are honored to have today Professor Luis García-Larrea from Lyon as a keynote speaker, and we'll have also as a speaker Neuroelectrics product manager, Pawel Skorupinski. Today's webinar will begin with Pawel's presentation with the duration of approximately 20 minutes. Pawel, during his talk, will cover the solutions for the home treatment with tDCS and home EEG monitoring. This will be followed by Professor Luis García-Larrea talk, also with 20 minutes, and then we'll have time for questions, so please feel free to submit your questions at any time using the chat tool. So, as you know, transcranial electrical stimulation currently presents an innovative research and treatment tool, which is an effective alternative to pharmacological and behaviorally based treatment. Currently, however, tDCS intervention must be administered over multiple in-person visits to a clinical or laboratory settings. It is therefore inaccessible to a large number of patients, subjects who live in remote settings or who are unable to organize transportation. And this barrier hinders the ability of a clinical trial and slows the pace of discovery and technology transfers of tDCS. So we propose to increase the accessibility of tDCS intervention to a large number of subjects and patients by using telemedicine and home-based tDCS administration. So now we'll start with presentations, and Laura will introduce the first speaker of today's webinar, Pawel Skorupinski.
Thank you, Rafael, and welcome everyone. So our first speaker today is Pawel Skorupinski. He received his bachelor's at Wrocław University of Technology in Poland, in computer science, specializing in internet engineering. He also received his master's at Dresden University of Technology in Germany, specializing in distributed systems engineering. He has worked with large-scale computing and resilient systems architectures in different corporations like IBM or Adidas, and he has also worked at startups. He is currently working as a product manager at Neuroelectrics, where he works closely with technical teams, and he also coordinates all the innovation work around our portfolio of brain stimulation and monitoring solutions. Pawel, the stage is yours.
Thank you so much, Laura. Thank you so much, everybody, for joining. I'm delighted to be here today and share with you some insights regarding our Starstim-Home technology. I will, especially that some of the capabilities we have just lately introduced. So it will be great to show you how you can use our technology today if you are interested to organize your home study with tES. So I will introduce in my presentation the home paradigm for tES study that we propose. And then throughout the years, our partners and first customers came to us with some of their questions and concerns. And in the following chapters, I will try to introduce how we responded to those concerns with capabilities and to those needs. So maybe you have similar questions. So I will go through how you can keep control and stay close to patients during the home study. Then how can you bring the lab study that you have chosen, that you have maybe designed, to the home settings? And then in the end, if you are interested as well in blinding your study, how you can make this happen. As a first part with a home paradigm, Our system consists of, this is kind of the telemedical architecture that we propose, that our system is based on. So if you look to the left, we have Starstim-Home kits that patients receive home. Right now, some studies don't even include the preliminary meeting of researcher and a patient. So it is a kind of self-contained package of components that are ready to be used by a study participant in home environment. Then on the other side, there's a research team that has the research team members have the web-based interface where they can manage their home study and as well access EEG files, as you will see later, and then can as well receive on their phones or any devices they have the real-time notifications, which we'll talk as well later. And those kits that are patients at home, as well as those web-based tools, are integrated. The data gets integrated in a institutional database that is in the based on the servers, and we can and it provides a secure data storage and processing. So let's start first from the left side. So from this home systems that patients or study participants in principle receive home. Starstim-Home is a hybrid tES EEG system. What does it mean? It means that it is an 8-channel device that on each of the channels can perform either tES or EEG. The device is compatible with both our portfolio of electrodes, which are either NG-PI stims that can do both tES and EEG, or more traditional tDCS electrodes, which are SPON stims in various sizes. Then we have cables that are optimized to help the patients with their home setup. And then we have tablet that serves kind of as an interface for the device, for the study participants and their supervisor at home to go through the entire session, that it's wirelessly synchronizing with the device and as well with the institutional database to synchronize the treatment. On the other hand, we have a researcher portal, which is an interface for research team members to jointly manage the home study. It is web-based, so these days it makes it also useful because it can be accessed and securely and directly, even when researchers are not in their office. So with that introduction, I want to go to the first kind of section first of the capabilities that I wanted to cover today, which is how can you keep control, how can you stay up to date and stay close to the patient throughout the home study. So there are a few concepts that you can find in our documentation regarding this topic, especially regarding the real-time functionalities. So I want to take this opportunity to explain them during this webinar. First of them is real-time remote access control that we offer. What does it mean? So by that we mean a mechanism which guarantees that the device can physically be accessed only during hours intended by the researcher. And this mechanism is managed remotely and in real time. So to give you a picture, let's go through through some demonstration. So let's say we have let's start from the US. Let's say we have a study in the US. Hello to everybody from America. We have a researcher in Nevada and participant in Ohio. Researcher has their portal interface and participant has their tablet that is also an interface to the device. So at the moment when the researcher schedules a session, it automatically synchronizes with the tablet. So that means that at the moment when researchers schedule a session, automatically patient can see that they have the session scheduled for a given period of time. Then let's say that this is a situation when the schedule is when the session is scheduled before actually it can be accessed. So here we can see that the session can be accessed between 3 p.m. and 6 p.m. So when the session start comes, you can see the tablet gets directly updated, and only at this moment patient can click the button to begin the session. And sessions in the portal additionally don't have to be scheduled one by one, but also the whole treatment can be pre-scheduled. And basically, as soon as the session is finished, patient can see when is their next session upcoming. So this is real-time remote access control. Sessions can be as well removed if the researcher made a mistake. Yes, and it gets synchronized with the tablet. Second capability that we offer referred to is real-time remote home events monitoring. So what do we mean by that? It's a fully automated mechanism which delivers information about study participants' actions and device behavior to research team within seconds. So it's our capability in our cloud platform. So let's go now to France. We may have a principal investigator in Lyon. We may have a researcher in Paris, and we may have a study participant in Bordeaux. So, of course, participant in Bordeaux has a tablet with a scheduled session, and researcher and principal investigator would have their portal access as well as their email inbox that they can access on any of their devices. And here, let's say that the participant clicks to start the session. This will automatically create an event that will get delivered to the researcher. So it's not a very important event, so only researcher would receive it. Now, if you go to another scenario, study participants chooses to abort the session. This will as well create an event automatically synchronized, and this will send an email to both researcher and principal investigator. And they will both will also have access to that information in the researcher portal. And additionally, after every session, each of the research team members has access to all the details start during this session. What were important events, as well as have possibility to download EEG data recorded during that day's session. So this is real-time remote home events monitoring. Regarding next section, how do I bring lab study protocol home? So you may have some specific study protocol in mind, maybe some requirements from your ethical committee, and you may be wondering how you can perform the same study that or very similar study that you would perform in the lab, how you could bring it home. So this session is about that, this section, sorry. Okay, so the way our home tablet software works, it consists of a step-by-step phases that the software automatically controls. So it means patient needs to go through some specific set of steps that are configurable, and we will introduce it here. So of course, as a basis, we have a tES protocol execution that with our platform, you can configure duration of the tES protocol montage. It can be optimized by polar 4x1 montage. You can choose a waveform, and as well a ramp up and ramp down time. And now this home software, always before patients access to that tES protocol, will reinforce automatic hardware verification at first, and then automatic headset setup verification. So only if all of those pass, patient is allowed to the tES session. Additionally, we have setup instructions and headset removal instructions to support the study participant with the setup of the device. and they are also adjustable for the language of the patient. So we have few languages available, but if we do not cover your study language, we can always allow you to kind of collaborate on translation for your study participants. Okay, so this is the basic tES session pipeline, and right now we have a few things that can be added on top of that. So we have questionnaires integration option. You can add questionnaires to allow study subjects to report on their condition or acute events. So session can consist of pre-questionnaires and post-questionnaires. And right now we allow multiple questions that can be mandatory or optional, and they can be single choice, multiple choice, or open. We also allow follow-up questions. So if patients answer that they have some symptom, then you can configure the question that is shown afterwards. Additionally, this is our latest capability. We have answer-based session access blocking. So it means, for example, if a study participant answered that they have a severe symptom, then our software will, if your team chooses to configure it like that, your study participant will not be allowed to access the session unless you take an action. Additionally, we offer the combination of tES and EEG with our protocols. So if you want to use EEG to monitor the safety with objective data on acute events, or you want to research neurological evaluation, for example, treatment predictions, then you can add to the tES protocol that our device would perform as well EEG monitoring. So this can be configured before, after, or during the stimulation, and you can adjust duration, and then as well choose the locations on the head cap where the EEG will be recorded. Further, we have as well allowed for what is being studied a lot with tDCS protocols, which is cognitive tasks. So if you want to perform cognitive training or treatment efficacy assessment with some cognitive tasks, then you can integrate in our home tablet the presentation software task that, for example, some standards like Go, No Go or Stroop, and or any software that your development team would like to integrate, and then just they need to always match some safety guideline provided by us, like allowing session abort from their screen. And then cognitive task can start with the EEG step or with the tES step. And finally, also we allow, if your institution has some video software available, we offer a possibility to integrate like a kind of video call button on our screen that patients can use to open that video software and call your research team straight from the tablet. Okay, so that's about this session pipeline design, and then the last question that kind of we have responded to with the design of our system is possibility to conduct double-blind studies. So our system is single-blind by design. So our we allow for automatic generation of sham versions of the protocol and adjustment of their parameters, and home patient software does not reveal any information that could lead to guessing of shamming. Additionally, we have double-blind configurability of the researcher portal. So that means that some of the researchers that access the portal can have the admin mode, and some of them can have blinded mode, and the researchers directly communicate with the patient will have all the information that can lead to guessing of shamming hidden from the interface. So this way you can assure double blinding. this was my introduction to the technology. Thank you so much for your attention, and I'm giving back voice to you, Rafael and Laura.
Okay, thank you very much, Paweł. We'll follow up now with the presentation from Luis, and then after the second session, we'll have time for questions and answers. Laura will present Professor Luis García-Larrea.
Thank you, Rafael. So our next speaker is Dr. Luis García-Larrea, who obtained his MD and Ph.D. degrees and a medical specialization in clinical neurophysiology from University of Barcelona. He is currently the research director at the INSERM Center in France. He is also the head of the lab Central Integration of Pain in Humans, Neuropain, at the Center for Neuroscience of Lyon. He is also a member of the Neurological Hospital Pain Center. He has served as a president of the French and the European Societies of Clinical Neurophysiology. He currently serves as a member of the European Task Forces for the Assessment of Neuropathic Pain and Neurostimulation Techniques. And his main research interests are the cortical processing of somatic sensations, the diagnosis and prediction of neuropathic pain, and its therapy using cortical neurostimulation. Luis, thank you very much for your time, and the stage is yours. Pawel, I think you have to unmute.
Put the sun on, that's all right. Hello everybody. And so as Laura said, my main domain is pain integration. So I will not be as technical as Pawel was before. Just we'll try to discuss with you some ideas about the possible mechanisms that are behind the tDCS systems and how to use this on a home base and why to do this in the context of chronic pain and chronic neuropathic pain, which is my domain. If I take my, apparently I cannot share my, I need the leader to authorize me to share my screen. Okay. Well, I hope you are seeing it now.
Not yet.
Not yet? Okay, you tell me.
Could you try again, Luis?
We cannot see your screen.
Yeah. Okay, I'll try again.
Okay, now it's starting.
Yes, now it's
working.
Yes.
Okay, perfect. Okay, so the idea is that we have gone in 20 years from something that started in the operator room to something that can be done at home. And in fact, the story is much longer than that. Cool. Okay. It's much longer than that because you can find during the 50s and the 60s already investigators who were playing with stimulation of the motor cortex. This is the motor cortex in the cat, and they were playing with rabbits, with rodents, and with cats, and finding that stimulating this particular part of the brain was able to inhibit the pain signals as down as the spinal cord. So the idea comes from very, very, very long, and it was started to be implemented for neurosurgeons, so implanted electrodes over the motor cortex in the 1990s by Japanese Dr. Suzukawa. So the first things that we learned from this is that contrary to expectations, when we were stimulating here in the motor cortex, things were not happening in the motor cortex immediately. This is a slide from a positron emission tomography done in patients that were being stimulated in the motor cortex. And the surprise was that things were happening, that there were activity changes in the brain, but relatively far away from the motor cortex. And this was happening, for example, in the thalamus. This is slides, no wonder the details, there are 4 different papers from 4 different groups showing that changes were happening in the thalamus when you were stimulating the motor cortex. This is very nice from Saito in Osaka. And we also learned that some of these changes in the thalamus were ephemeral. So they lasted just during the stimulation time. But there were other places in the brain, here, for example, the orbitofrontal and the perijuvenal cingulate, that could last much longer. And in fact, they may last and may be essentially active once you had switched off your stimulation. And this was a thing that nobody was looking for, that the real thing that was happening in the brain, and in tDCS is probably the same, is after you have stimulated, during the hours that follow the stimulation. And it was discovered that these things were happening not only in places far away from the motor cortex, but also in places that were interconnected between them, not only anatomically interconnected, but also functionally interconnected when you play with the curves of activation of it. So they were a kind of sequence of activation that started here in the motor cortex, but that very rapidly went away. And the effects against pain were most probably not driven in the motor cortex, but driven from this very huge network that was being activated. So this is more or less the model we are playing with, and this is a model of motor cortex activation that has been reproduced in animals, has been reproduced in rodents, has been reproduced in cats, and that it seems to be able to predict the effects of this technique. So now, the question is whether these models that have been derived from epidural stimulation, which is phasic, and has been applied later to transcranial magnetic stimulation, which is very much alike the first, we are not going to speak about that today, whether they can be also applied to stimulation by direct current in the, over the scalp. In fact, we do not know. But there are things that are more or less reminding of that. First, that also this activation by direct currents can be very much longer and can go much longer than the actual duration of the stimulation. And this was also found many years ago. This is a paper from '64 showing that there were very long-lasting after effects of this technique. This is just the actual recordings. Another thing that is very much alike is that now when we do the first experiments with functional imaging during tDCS, during transcranial direct current stimulation, we also tend to find that the activation arrives in a very widespread array of structures that are far away the motor cortex where you are stimulating. So this notion of activity that is disseminating in the brain stays also for tDCS. And tDCS has a particularly that unless you do it with very close arrays of electrodes that ensure a very, very focalized activation, if you do it on standard grounds that many people are doing, it is with an anode in the motor cortex and a return current from frontopolar, for example, then the distribution of the voltage is itself very large. So this is not just the activation of other parts, this is just places where the voltage is going when you put an electrode just here, over here. So you see that putting an electrode over the vertex, you have a non-negligible activity, excuse me, not an activation, but a portion of this voltage can arrive to the posterior insula, which is very, very far away. But the posterior insula is important because this is the place where pain influxes are arriving. So this is a characteristic of tDCS that differentiates from other techniques, either epidural or RTMS. with tDCS, you can also play with the extent of the current that you are applying as a function of the distribution of the electrodes that you put onto the scalp. This is just another example. And still, one commonality with other techniques is that you can, with tDCS, modulate the level of neurotransmitter in distant areas. And this is a paper appeared 4 or 5 years ago, when they explored the brain metabolites by doing fMRI, but with a spectroscopy fMRI. And so they could verify that there were changes in the level of glutamate, which is an activatory neurotransmitter, but also of GABA, which is an inhibitory neurotransmitter, in different places of the brain, just by applying standard tDCS. So we have commonalities, and the commonality that interests us most is that this is quite recent, it's from last year and from this year, this activation of the motor cortex by tDCS particularly modulate pain that is pathological rather than just experimental pain. In fact, it is not very good to modulate experimental pain, but it's much better to modulate pathological pain, such as the one that we induce with capsaicin, for example, generating hyperalgesia, with other pain models that try to mimic a pathological pain. So we have something that seems to work, and when it works, we try to do with systems more or less sophisticated, perhaps not as sophisticated as those of Pawel, but just trying to have also questionnaires in double-blind or single-blind system where the patient itself notices every day the evolution of the pain. So this is a week of stimulation standard with magnetic stimulation, and this is the same patient with the same type of pain, of course, but stimulated with tDCS, and you see that in this case you have a nice response from both techniques. This is not always the same, but we found many patients that can respond to both techniques. So if we want to be pragmatic, whichever the technique you wish to do, you need unlock some of the difficulties. There are many, so we'll not be speaking about all this. We'll be speaking about the last of this. When a patient is sensitive to this technique, your first problem is how to implement a method to sustain the benefit. And this is almost impossible in the hospital. You become a kind of hairdresser with people queuing at the gate, just coming to come again and again to have 1 day or 1 week of stimulation. And this is why we started to use the systems able to perform at-home stimulation. At-home stimulation is not the only way that is being developed to ensure long-lasting effects of these techniques. There is another technique that is being now getting more in fashion, which is what is called slow pace. It may be a slow pace RTMS or slow pace tDCS or slow pace whatever. It means to make sessions repeated only every 2 weeks or every 3 weeks, because we know now that because of what I showed you before of this kind of effect that is delayed after the stimulation, we can create a kind of cumulative effect of whatever you are doing for this type of stimulation. So little by little, you can have your patients being better and better just with 1 session repeated every 2 or 3 or 4 weeks. The second way, and this is the way which I'll be speaking today the most, is rendering the patient autonomous, so making the patient the person that stimulates itself. Pawel explained it very much better than I can do, but essentially what you have is a server in between, and then you have on the one side the doctor that is authorizing the session, on the other side you have the patient, both connecting to the same server with a very much control system that does not allow the patient to stimulate him or herself whether it wants, but just at the moment that we have chosen, with the technique we have chosen, and with the parameters we have chosen. This is very, very important. And then it goes. This is one of the paradigms that we are using. The first thing is to have a baseline. So the patient must establish during at least 1 week, sometimes 2 weeks, the baseline level of his or her pain, and other qualities. It may also be as a function of the questionnaire that we send, also the sleep quality, or the fatigue, or the quantity of drugs he or she is taking, etc. But we need a baseline. Then what we need is a training. The patient should be trained to do what he should do. In our case, we do it in the hospital. So the patient comes 2, 3, 4 days. It depends on the cognitive status of the person, just to learn how to do it by yourself. At the end, there is a kind of mini exam. So when we want to be sure that he is able to do this autonomously, because otherwise it becomes very much complicated. And then the patient goes home to repeat the stimulations on in our case is during 1 month, during 4 weeks. Then after which he continues by daily explaining, either on a paper or on the internet, their ratings labeled day by day, until, at least in our case, at least between 80 and 90 days. So we try to have a very long pace afterwards. He goes with a kit. Well, I'm sorry, because this kit is much less advanced than the one that Pawel showed you. This is the previous kit, but we are poor, and we are on this one. We are now moving to the next one, but the essentially is the same. So the patient has a tablet, or a mini computer, and he goes with a stimulator, with cables, with electrodes, with gel, with everything that he needs, and everything takes in a very, very small mallet like this. Then he learns to put the cap, to put the electrodes, and to put the stimulator, and to stimulate. This is our last patient. This comes from 3 days ago, so this is why she's with the mask, and she's in fact not really training at the hospital, because this lady is the 4th time that she takes during 1 month the stimulator, and every time she gets 6 months of pain relief by 1 month of stimulus. So this is quite interesting. She's a young lady with hemiplegia. So she's really tough pathology, is thalamic syndrome, and she's doing this about twice a year. And with twice a year and some drugs, she's able to keep the pain at a low level. Now she was very much unhappy, because during all the confinement, she was not able to come and to reproduce the stimulus. Then when you do it, well, this is one of the things that you can obtain. This is a good patient, if I might say. This is the level of pain at baseline during 1 week. This is the week of the training. And when we do a placebo control, which is what we are doing currently, this placebo, we introduce it at the beginning. Because of a very, very simple reason, is that we do not know which is the post-effect of the active tDCS. And this post-effect may last sometimes weeks or months. So if you introduce the placebo just in the middle, you don't know what you are doing and what you are receiving. So we have only the possibility of either doing parallel groups, which is not ethical in our, because we need that every patient be treated, or introduce the placebo at the beginning. Okay? And then we tell the patients that it was a placebo just 6 months later. Okay? This is a good patient. So this is a bit more complicated, because you see that the stimulation is here the placebo, here's the active tDCS at home. During this time, the benefit is really small relative to the beginning, and you have to wait. I don't know whether you see in my screen, this is a bit close by the heads of my colleagues, but it is just at the day 63 and more when the VAS becomes about 5 versus 7, 8. So sometimes you need to wait for really several weeks until you get a good data. This is a kind of group analysis. If you look to the higher part of the screen, these are all the patients that we have recorded so far together. So you see that these are the standard deviation relative to the baseline. This is the sham stimulation. This is the active tDCS, and this is the continuous rating till day 77. What you see here is when you split your patients into two very simple groups, those who have more than 3 standard deviations from the baseline and those who don't. You see that you have the non-responders there and the responders here. And what is important is that there is no or almost no overlap. So you have patients who respond and patients who definitely do not respond. And if they have not responded after 1 month, in our experience, they will not respond. In our hands, this is about 50% of the patients. So 50% of the patients responding, you may say is more or less, it depends on your experience. If you take into account that these patients are all chronic pain patients with neuropathic pathology who have not responded to any drug in the market, to be able to have half of them and provide some benefit for us, this is a clinically relevant point. So now before ending, because this is very nice and this is a bit too nice for you, so I just will show you some of the problems that you may find when using this kind of technique. One of the very classical problems is that the pain, chronic pain, is very proteiform. So you may have a continuous pain, you may have paroxysmal pain, you may have allodinic pain when you touch yourself or you to something, and these different types of pain may not respond in the same way to the technique. This is a case with a patient with essentially painful paroxysms after plexus avulsion. So this is very, very much painful. This is the evolution of global pain, and you see that it's not very much important. So it is a very slightly improved during some days, and then it comes back to the beginning. The continuous pain does not change very much. The intensity of the paroxysm does not change either. But this patient was extremely happy with the stimulation, and he has used already 3 times, because the frequency of paroxysms was very much decreased. It's a bit like having epileptic seizures. You may have 20 per day or 2 per day. Even if each seizure is as important as the others, you only have 2 per day. And this was very, very important for him. This is what he wanted to continue the stimulation. These things you have to think about it. So the questionnaire should not be just which is your overall intensity of the pain, because you will not get the good responses. In general, you should need to have at least 3 axes. One is the global pain with all the categories, paroxysms, intensity, frequency, etc. The other is the drug intake. If you arrive to make your patient take less drugs, then you are being objectively useful for him. And of course, the other is the quality of life. In the quality of life, you use whatever type of questionnaire you wish. There are plenty that have been validated. It is extremely important because without this, excuse me, I lost my thing. Without this, you can lose very much important information, like this one. Okay. You may have patients that have neuropathic pain. When you establish which is the global pain change after 1 month of at-home stimulation, you get just a 70% of improvement, which is barely significant. But this patient comes to you with an enormous smile and wanting to do it again. Why? You don't know. You only know that if you don't investigate the quality of their life, you cannot investigate why are you being of benefit for these patients. This is another case, also with 50% pain decrease, which is very modest, but these patients could do the supermarket. This patient could go biking again. So even this 15% decrease may be extremely important for the patient. So do not get annoyed just by these figures and try to include always your quality of life. Okay, I think this is all that I had to tell you. All this is now. I just wanted to end up with all these people that are working in the lab or at home now at distance, but they are the responsible that I am here just to talk to you. Thank you.